So welcome back. So in the first session we heard about the general principles of DREAM from Gustavo and some of the practicalities. And Brian introduced the platform Synapse. And then we thought what we could do next is to go over a challenge where you guys will be able to play and get all the excitement and so forth involved in being a participant. And so what we'll do now is to present the challenge and make teams. We can discuss briefly the best way for this and then play around. So what I will do next is to introduce you the challenge. So we thought quite a while which challenge to use. As Gustavo presented, the challenge can be, they started network inference, but then we have done different challenges like predicting drug response, predicting disease prognosis. And then we decided to choose a challenge that has a bit of everything. It's mostly around the network inference problem, but it's in the context of cancer, and it's a fairly recent one, so we thought it's a nice example. And so it's part of a challenge that we did 2 years ago. And I will tell you actually after you do the challenge more about the real challenge, because if I tell you too much now, I will bias or I may influence your participation. But so just a few words about what the challenge was about and what our challenge today will be about. So again, as Gustavo said, reverse engineering was the starting point for DREAM, and is a main question in computational biology, and that many of us are working on that. It was addressed in DREAM, and Gustavo showed you the wisdom of Krauss paper, so the Marbach paper. We also look at this in the context of signaling. So Marbach was gene regulatory networks, and the other one was signaling pathways. And a bit following that challenge that was done a few years ago from data from Peter Sorger lab, where I was as a postdoc. A few years ago, we did one challenge called the HPN DREAM Challenge. HPN is Heritage Provider Network, who was the sponsor of the challenge. And data came from Joe Gray's lab. But the question was to try to identify causal links in a signaling network. And this data of signaling data came of cancer cell lines. But first about the problem, right? So why it's important to look at causal links, so as opposed to just trying to find for correlations, right? So the idea is, can we find links that have a directionality and a sense of causality, so that if you have two nodes, say two proteins A and B, we want to find the links that are determined by the fact that if you affect the protein A, you block it, you destroy it, you should see an effect on protein B, right? So there is a causality. And this is particularly complicated. It's even more complicated than infer networks of correlations or networks of relationships. And is confounded by a number of aspects. One of them is that these causal effects can be direct or indirect, and it's very hard to tease this apart. So for example, if you have two proteins like here, it may be that the effect is direct from A to C, but it could be that there is an effect from A to B to C. And so there is a number of important problems, and that's what we tried to do, is to pose a challenge where we could try to identify this type of causal links. So a bit going beyond previous challenges, which were more on finding undirected relationships. And as I mentioned before, this was in the context of signaling. So is everyone familiar with signal transduction, signaling networks? So I need to tell you more about this. So we look at signaling pathways in breast cancer cells, and our system connects receptors all the way down to transcription factor involving kinases in between. But since it seems everyone knows what signaling pathways, we just skip this. I mentioned that in the challenge, and I will come back to this after we do the hands-on, we had real data coming from Joe Gray's lab, and in particular, Laura Heiser was the main person providing this data. And this data was in breast cancer cell lines. And just to give you a flavor, we had 4 different cell lines, 8 different stimuli, 45 measurements. So this is phosphoproteins, which more or less tell us about the activity of proteins. And these cell lines were stimulated with ligands affecting or activating receptors, but also with inhibitors blocking the activity of kinases in the network. So we had a very rich data set, and then what we tried to do is to ask people different questions. So we split the challenge in sub-challenges, and we will do the same today. The first sub-challenge was, can you tell us the underlying causal network? And the second sub-challenge is, can you give us prediction of some of these proteins under some of these conditions? So that was with real data, but also we did a challenge with in silico data, which is what we will use today as the basis for our tutorial. And this in silico data tries to mimic the data I showed you before, so it's a signaling pathways of relevance in cancer, but come from a model. It's not real data, it comes from a mathematical model that was used to generate data under similar conditions, namely different ligands activating pathways, different inhibitors, measuring 20 proteins over 10 time points. And the questions are the same. Can you infer the network, and can you predict the time course? So why do you think we did the in silico data if we have the cool real data? What is the reason we did this? Robert, you cannot say. Any of the other ones? Any thoughts? Exactly. Because, I mean, okay, for trajectory prediction, you have real data, but the ground truth of the network in real cases, in general, is not known. You have, as Gustavo called this, like silver or copper or whatever. I mean, there was a nice case, actually, where there was this basically synthetic biology build network, right? There was a few years back from in Dream 2. But otherwise, we don't really know the true network, right? So then often we try to combine real data with in silico data. The advantage is in the silico data, you control your system, you know the ground truth. The disadvantage is it may not reflect the real case, even though, of course, we try to put assumptions and build into the model things that mimic as well as possible the real data. Good. And I will tell you after where this comes from, but not for now. So for now, the data is anonymized. So we don't know which proteins, which pathways are the real or this data is derived from. But in abstract terms, so we have two ligands in different concentrations, low and high, that we use to activate these pathways. Then this is combined with inhibitors, as I mentioned before, in a layout like shown here. And then we have measurements for 20 proteins, also anonymized. And some of the drugs affect some of the proteins for which we have measurements. And similarly, some of the stimuli affect some of the proteins for which we have measurements. So this is the data. And then the data is stored in a format that we call Midas. It's something we developed also when I was in Peter Sorge's lab. It's a simple form to encode in a spreadsheet, in a table, data collected upon perturbation. And the idea is that in each row in a matrix, in let's say an Excel or comma separated value file, you would encode a specific experiment. So each row is an experiment, and then in columns, first you encode the conditions of your experiment and their different labels that start with TR for treatment. So for example, treatment inhibitor 1, and then in a given experiment, let's say row 2, for each of the treatments you will annotate whether a specific condition is present. For example, if a certain ligand is present, you put a 1, or if a certain inhibitor is present, you put a 0. And as soon as I finish this, we of course can open these files and take a look together. Then after the
Midas was minimum information for determining
for data analysis in systems biology, something like this. Yeah.
So the gold standard.
Exactly. Yeah.
Midas.
Exactly. The Midas of systems biology. But it's just a convention. Doesn't, of course, it just helped. The reason we use it is because a number of tools that we have developed can easily process, plot, and transform these data sets. So after defining the conditions, you would define the data acquisition time. Sometimes you have measurements, so you do experiment, let's say you put a drug, and you may do different measurements at different times. In our case, it is for all the measurements at the same time. Even though I said before that we have time points data, each time point comes from a different experiment because it's a different well that you measure from. And then what you do is you encode those in different rows to make clear the fact that it's a different experiment. It's not from the same experiment you measure different times. And that's why, in our case, each row is an experiment, and its time point is a different experiment, right? So they all have the same time, but you will see many rows in the matrix. And after this, we will write for the different antibodies that are reflecting the activity of proteins anonymized. For 20 of them, you will have the actual measurements. So this is what data will be available, so the format of data. And then also to tell you a bit more about formats, because we're going to build networks, and that's what you have to submit, we need a format for these networks. And then what we did is to use the Cytoscape format. Anyone does not know Cytoscape? So you'll know Cytoscape is probably the most used software for network biology. And then it has this format called CIF and EDA. So CIF simply reflects the architecture of the network. You say, for each edge, you write a row. You say from where to where. And if it's a positive effect, you put a 1. If it's a negative effect, you put a -1. And then there's this other format, EDA, which is like for attributes. And here, what you more or less, you expand the CIF by putting an attribute, which is a number. And this is important because we will encode here, or you will encode here, your level of confidence in a given link, right? So you may know, okay, this link is there and I'm pretty sure, so maybe I'll give it a 1. Or from others, you don't know, but you still want to tell us that you think possibly there is a link from A to B, but you are like only halfway sure. And this is important because we will use it also in the scoring, right? We will assess based on this. And then, so this is what you will provide for the first sub-challenge, which is the network reconstruction challenge. But the second sub-challenge is prediction of data, of trajectories. And for this, you will use, again, this Midas format I mentioned before. So the same way, and the templates are available, so you will only have to fill on the right where the question mark the values. So this is the 2 sub-challenge, what you are expected to submit. And then we thought, of course, you may want to try your own methods. Maybe in the next hour you come up with algorithms or different approaches. But to facilitate the participation and also to give you some baseline methods, we are going to use, or we have available, the actually best performer methods from the challenge when we run it. These are encoded in Cytoscape again, in Sinai, which is a plugin of Cytoscape developed by Oriol Guitar-Pla and Beno Stokowski in Paris and at Pasteur Institute. And then actually as part of the real challenge, there was an agreement with the Cytoscape team, with Trey Idekar and Ben and so forth, that they would encode the best performing teams algorithms, and this has happened and it's available. I'll tell you maybe later, after the talk, a bit more about the best performing teams, but what happened is that we had the best performing team for the in vitro data and one from the in silico data. And then that's why there are 2 methods available. I mean, the latter was actually globally the best one, if you consider both sub-challenges. And then, so you can run this or you can run your own methods. Try what you want. And then, so as I said before, we'll ask you to provide a network. This is sub-challenge 1 that will score using some erosive curve approaches, but we don't say too much now. We can discuss this later after you have participated, and Pablo will talk a bit more in general about the scorings. And then for the second sub-challenge, we will just try to score how far off your predictions are from the real data. I mean, real data means coming from the model, but there is data that we have, so we have withhold it. And then we will see how far off you are from here. And here we will see how far off you are from the network, which in this case we do know because the network comes from a computational model. And I just wanted to give you a flavor of this. As I said, we can go over this in more detail after you have participated. But just to thank everyone involved in the challenge. So as Gustavo said before, and we'll talk about this in the last session, building a challenge is a complex endeavor involving a lot of groups. Here, the data came from Sach and Paul Spellman and Joe Gray's lab, in particular Laura Heiser. The in silico data that we are using here came from Heinz Koppel lab, in particular Michael Unger, his student. And yeah, so this is the people that made this challenge happen, besides all the participants that were really the important people. And as I said before, this was funded by Heritage Provider Network in the US. And that's really all I wanted to say for now, just to give you a bit of background. But before we start, do you have questions? So if not, what we can do is we can really open Synapse and start to show you and look together at the files and stuff. What is at best? I do it from here. Brian, what do you think? I should...
Before, no, because you just came, right? But you can maybe try to register the Synapse.
If you have a Google account, it's super quick, because that's probably the fastest if you have one. Or
it's fast regardless of which one.
Yeah. So this is me in Synapse. So we built a Synapse page with Brian's help about the challenge. So whenever there is a challenge, there's going to be like a page with all the different aspects and all the documentation that you need to know. So here we did one taking from the real HP and Dream challenge most of the information, but adapting it. So if you were to participate, you will really get a lot of information about the challenge. We can take a look at this in some.
One thing I think we can say is that now, I mean, after a few years of doing this, now really all the challenges look like this in Synapse. So it's really kind of like a
standardized
template, and whatever kind of challenge you organize, these are the things that you have to solve. You have to figure out before you start the challenge.
What are the questions?
Yeah, so it goes from
describe the data,
description, question, timelines, incentives, right? And incentives, as we said,
a beer.
A beer and pizza today. Yeah, maybe we should.
A surprise prize will be given to the best performing teams. No anymore a surprise.
I guess the other things we haven't talked about, leaderboards, right? That's also very important.
Yeah. So they're here.
So basically, the idea is, there are, I guess, several things we want to do is, first of all, be very clear and have the data as available and as clear and clean format for participants, right? This is, I guess, one of the main things of Dream, right? In general, it's very hard to go and get the data somewhere and compile, download it, something. So one thing we do is make it as simple as possible. The other is, and this is to attract participation, right? Because we want these people to participate, and the more the merrier. So that's why also incentives are important, and that's why even leaderboards are important, because leaderboards basically make this thing very lively. You see people submitting results. Hopefully there's not right away someone who's just rushing all the rest, because that really hinders competition. But if you can send solutions and see them happening, it really attracts. You really feel part of a community. So this is also the idea of having a kind of a template for a challenge that any kind of challenge can be described with this. For example, this year I organized a challenge on predicting olfaction, strong structures of molecules. It's a very different challenge from the one we're kind of playing with, and it's also a very different challenge from the one that's being organized in trying to predict ALS outcome. It's more kind of patient-oriented, but all that kind of fits in this format and this idea of competition with a leaderboard and like Julio did, and really in basically 3 lines you can understand what is happening.
Over the years, over the different challenges, have you found that each challenge creates its own community, or that you have people stick to the concept and just whatever the biology, whatever?
I think really it has been, it has gone both ways. I mean, there are people that just participate in every challenge. Definitely, for example, I think the olfaction challenge, and this is kind of the part of being direct, is trying to get new types of data, so new fields, tap into new fields. That really has created a new community, because in olfaction in general, there's not so many data sets available, right? As in genome sequencing or gene expression or protein-based interaction. So I think if you can, and that's also very important for new data, and good data, that creates new communities and attracts new kinds of people.
But another side of this answer is, we don't think, I'm not sure that we succeeded in creating communities. I think that there is people that are following different new challenges and participate. There is a coincidence of interest that maybe they find the data interesting for their methods and so on. I wouldn't say that we really created a community in the way that we imagine that maybe would be, condensing around the topic.
So I wonder if we have the discussions that they kept recorded, because they are nice discussions. Maybe we can try whoever say. I mean, we had a mic, but we don't.
Oh, yeah, there's.
I was wondering, but this is recorded on the laptop, so maybe next discussion, whoever comes here and talk. But yeah, so what I can show you now briefly the file. It's more or less what I explained you before, right? The different conditions. You see how there are many of them, and then there are all these measurements. But the nice thing of the Sinai, the Cytoscape plugin, is it will automatically recognize the format with one glitch that Pablo can explain. We need to transpose the matrix, but so we can basically use that maybe as a starting point. But before that, so how do we want to make the teams? Because the nice thing being small group is we can maybe have some of us partnering with some of you, and you are at the moment 1, 2, 3, 4, 5, 6 participants, and Robert, we get 2 groups. I think that's good. 3 and 3 and some of us. Okay. Does it sound good, like 2 groups? So the good thing is, you already got the silver medal, right? So let's do that. Any preferences, or we do like by where you are sitting? That's the simplest. Yeah. Well, you go. It's 2 and 2 on the last row. So
who has experience with the working space already?
That's a good one.
You have. Yeah, they have.
The 2 of you have.
Oh, yeah.
But you have thought a little bit about it. So I do something that you're looking for. Why don't you divide?
Well, but you said you too, right?
Not really.
Jam, you do have.
Yeah.
Not count up one.
Yes.
Okay.
So maybe put them on this side.
So
Whatever.
alphabetically. Okay, so I will choose. Let's can change it. Jame, Stephen, and what's your name? Estela. Estela. And then Eugenio and Xavier. It's okay. And Robert has a little experience as well. I don't know, maybe you have to.
Yeah.
All right. Okay, so
let's move around.
Yeah.
And so then Brian will show you a bit more how to operate with Synapse hands-on.
They're building two rows faster.
Sorry, too.
2 rows for the 2 teams.
So once you co-organize here and log into your Synapse account, you can create a project to store some of your work.
Let's wait until they are set up.
Okay.
So you will need a different computer each team.
Okay, so the back team is the front team. Jaume is a Catalan.
He's a Catalan.
So you're from Catalonia?
Yes.
Ah, okay.
You were born in the Catalan.
I was born in Capital.
Do you?
No.
Casi no.
Es que Cataluña es
So, okay, so you guys.
Yeah, so he's gonna just show you, I think, I guess, the basics.
So Brian can show you now how. So you all are on Synapse? You are on Synapse?
Yep.
You guys are on Synapse? Yes.
Great. So if you have the link to the overall project, the workshop project page, as Julio mentioned, there's a lot of sort of meta information that you can go through here. And then as far as accessing the data, click on that wiki page, and that will link you to where to download the data. The link was broken about 5 minutes ago, but it's not anymore. And so you can go download it. And then as far as organizing your results and submitting to the challenge, if you go to your profile page, under the Projects tab, you can create a new project right here. And that's where sort of you'll store your submissions. I don't know if we'll do it today, but you can provide a write-up of the type of method that you may have used. And instead of creating one, I just created one this morning. I'll show you sort of what it might look like. So I created a wiki with methods, results, and conclusions. You can see this is a demo project, but showing that you can include equations in here, including LaTeX, some results and your conclusions. And then under the Files tab, I have 2 folders, one for Sub-Challenge 1B, and actually I have an example submission here. So if we drill into this, go to this Files page after you've uploaded your submission, you can go over to the Tools menu and click Submit to Challenge. And this will bring up, these are all of the challenges that I have access to.
You will have only one.
You'll likely only have a couple after you've registered. But you just select which challenge it is. You can alternatively provide a submission name. Go to Next, and it says your file has been submitted. Please watch your email for updates regarding the validity of this submission. So what we have is a harness behind the scenes every 2 minutes taking a look at submissions that come in. And there are 2 steps. One step is validating that the submission is in the right general format, so that it's a zip file with the correct number of files and the correct format. So you'll get an email initially saying your submission is valid, it will now be scored. The second level of it is the actual scoring algorithm, and once that is done, it will populate on the leaderboard of the wiki page, on the challenge page. I'll show you another way that you can submit. Instead, if you go to the wiki page of the workshop, there is a sub-wiki for it's ISMB AKES and then it should be sub-challenge 1A or 1B. But you may not have registered for the challenge yet. So the first thing everyone needs to do is to register for the challenge.
Are you guys registered? Or do you want to see
Are people registered? Yes or no? Yeah. Yeah, if you go to the workshop page. So you go to registration, the registration link, and there's information about how to get a Synapse account, how to become a certified user, and then at the bottom there's a join challenge link. Yep. I can help with that too.
Team front is already in?
I sent that request, but
You do the become certified user before. Did you answer the question?
Okay.
You do the become certified. Yeah, I did it.
you have a name and it's called
So another way to submit solutions, there's a submitting solutions wiki page here, and you can submit to the challenge. And here it will give you only the 2 for this challenge. And you just pick which one you want, and then you search for the entity, your file.
Either from the file, you go to the challenge, or from the challenge, you go to the file.
Then can we see, register, but can we see the team, the challenge team? So they should show that, right?
I don't think we set it up to do that for this one. It's a configuration thing. Yeah, yep. And we didn't know with how many people we had what we'd be doing. So. All right. Straightforward enough for now.
Are you guys good? All things are. You got the settings or not?
I can grab it. I'll grab it from my computer over
Okay,
here.
give you the settings in a moment. And click back in.
So you need to make a team.
So in the meantime, for you guys, I mean, what we suggest, as I mentioned before, is to use this Cytoscape plugin. Do you have Cytoscape installed also? Good. And the app, the Sinai app? Perfect. I mean, I guess you can already download the file. And Pablo just left. But there is just a trick we need to do, which is so because the Cytoscape format is the inverse of this Midas, so the columns, but rows are columns, columns are rows. So that's the only thing you need to do to just run Sinai. And then you could try that as probably the first step is to run Sinai with one of the methods and submit, and then you have like a baseline. I mean, the baseline is the best performer, so.
If you can do better than that would be great.
Yeah. But in Sinai there is also an Arachne-like method, so you can try a bunch of methods. And so maybe I can open Cytoscape.
There is an X in Cytoscape.
At the same time.
Yeah,
With almost join forces.
if you have any questions,
We go there. Huh? So. So I have the Sinai Toolbox here, and you will see you have different algorithms, like mutual information based, like Arachne kind of Bayesian. Where is the It was Pablo who was looking at this. We should grab him back. Do you know where he went? No. Okay. So it should be here. Yeah, Pablo ran both methods with the data to get them to Brainstorm. Maybe I have another version here. Maybe that's it, because I haven't uploaded my version. Like a newer version. Let's see the. Yeah.
I really don't know whether it's something that you have to choose from the possibilities.
Maybe the next time we can start to think how would you try to find a network from the data? Right? You have this I personally didn't think much about the method, but so we have a bit of time, and that's really part of the challenge is really think what is the best approach to solve the question, right? So the question is, you have measurements for nodes over time under different conditions. How can you find from that information the underlying network? I'm here, yeah, fine. Let's have a chat later. That's great. Yeah. Great. Later. Well, here's the Sinai guru. So the first question is the methods. And by the network, the file they have to transpose. You want to drive this part because you are the expert. The file is unload, yeah. It's on the download folder. You have to load the data.
Where is your file?
MD silico in there. MD in silico. MD in silico. So that's how you would load the data.
It's CSV.
Yeah, CSV. We have to click select table data to apply algorithm. You click from this one.
First step. Is it?
You want to look
Yeah.
down here.
Let me just put some.
Yeah. You can move this.
Yeah.
55.
Yeah, I know something. Okay. Okay, well, I can do
So in the meantime, maybe just to reflect the process. So in the challenge, right? So as I was saying, so you were given this data and you're asked a question, and the one part, which is arguably the most important one, is to brainstorm and think what is a good approach to solve the problem. The problem is you have measurements for proteins over time under different conditions. How can you reconstruct from that data the underlying network? So Pablo is going to try some correlations approaches, so what other methods do you think one could try to apply? Okay, so to principal components to compress the data before running the. Okay. Yeah, in this case, it's molecular data, right? So the phenotypical data is not there, and so.
There has to be both processes where the surrounding the neighborhood of that mutation is a graph of the
And if whatever comes up with something that's technically you've seen before, it will come to this.
That's for this. So you didn't find it.
So in the meantime, so to comment, right? So another thing that you could try to use normally is to bring in other sources of knowledge. But that's not the case here because it's all anonymized. In the real case or in many challenges, right? Because it's real data, you can say, for example, I look in databases for what is known about these proteins. So in this case, and also we chose this to keep it simple, so it's just the data. But another place to look for information is prior knowledge. And then, yeah, there are many different inference methods, also based on differential equations, because we have time series. You can try to fit models to differential equations. You can try to. Okay. No, we do have time series. It's 10 time points. And this will be in the file.
Well, I think that is like they ask where do I see changes first? Give me something that is normal.
Yeah.
Then I ask hey, so there's any changes to the base change.
Yeah. That's good.
I'm going to try to get mutual information that is the lower threshold.
Sure.
Just I sent Julio my data file, which is the one that works, so I wanted to try with that one.
But maybe it's because it's not transposed that way.
Maybe that's why, so I want to just check.
Yeah,
So I just sent it to you. Okay. Yeah.
So maybe what's the best thing to? Can we upload his file to the Synapse quickly? Maybe that's the simplest.
Yeah.
So just to speed up things, we will do the data massaging, or Pablo has done this so that you can run this right away. But this phase is like brainstorming. I mean, I think it's good that we are doing it also here because it's really critical. So. And what you were saying about early and late is actually one of the best performing teams did something similar. So. So Pablo, I upload the files you said. This is your file.
Yes.
Let's just first apply correlation because it's just the same.
So this is the file formatted for Sinai. Well, it's still not work.
Look at the
You can.
well actually. I think. Oh, he just put it.
File?
Well,
No, I downloaded
sorry,
it.
that's why. Okay. Oh, so you just downloaded. You haven't uploaded.
I did not. Yeah.
Okay.
It's loaded.
So I have to load it. Sorry.
No, I did that already.
So you loaded?
So one of it. Yeah.
So this is one. This is in silico, right? This is.
I mean, you can upload again.
I think it's this one. That's
Well, it's a file that he sent me by email. Do you want to just to be sure? Go to Go there.
yeah.
In silico.
This is the one.
Okay. This is a new one.
Yeah.
And I say okay. Maybe I should have.
I think you have to. You have to play a little bit with this.
So we have a file that the one from Pablo. I already did. Yep. So this is the same place? So when you see new files download, okay, let's see. You can download the second version of the file if you want. But you want to, I mean, if you're happy with everyone,
I don't know why this is so blue.
yeah.
Wait, let me just. Yeah, do you have the? Can you just bring it back?
What do you need?
No, I mean the.
Are you close?
Yeah, I just closed it. No se puede ver aquí, ¿verdad? Está filmando. And outside.
For this part, maybe you can.
Okay, so now let me just.
Maybe we can upload once when we do try.
Yes.
So you ask, how will you like
So this is a
you guys are familiar